last 5 or 6 years, working together with a very dedicated team, is develop a test that utilizes saliva and can accurately distinguish whether a child has autism spectrum disorder or does not. And this relies on a new type of molecular analysis that really wasn't even possible until recently. We've published actually 5 different studies to date on the specific use of saliva for accurately diagnosing autism spectrum disorder. We had some initial pilot work that was published using small samples. It was really a proof of principle. And then we have had a number of different publications since then, stretching back almost 6 years now, where we've looked at different classes of molecules individually and reported on their ability to differentiate children with autism from their peers. Last year, we published the culminations. This was the largest study involving approximately 450 children. It was published in a peer-reviewed journal. It was very thoroughly reviewed, and it was also very warmly accepted, we were delighted to see, by our scientific and medical colleagues, who were very surprised to see that saliva could be used to accurately distinguish children with autism from those who did not have autism, and that there was science behind it that backed it up. Saliva, it turns out, is a treasure trove for molecules that might reflect brain function. And we felt that was a key component for the development of a test that's going to essentially be evaluating a change in brain function. The reason that saliva is so well characteristic or reflective of what is going on in the brain has a lot to do with the simple fact that the brainstem innervates so much of the oropharynx, where saliva is made, where you swallow saliva, where you speak, where you encounter the foods that you're putting in your mouth all the time. It became very apparent that the molecule types we were interested in evaluating were actually present in saliva at levels that was higher than you find in blood, in other biofluids, and in other tissues. And what was there in saliva was indeed highly reflective of brain content or brain contributions to the oropharynx. The reason it's not a genetic test is because we're not assessing or looking for a change in DNA sequence. We are specifically pushing a technology that utilizes epigenetic testing. We believe that the epigenetic level of testing that we are providing represents a final common path. So whether a child may have developed autism spectrum disorder as a result of one particular DNA sequence change or another one, or potentially some environmental exposure that pushes them toward autism spectrum disorder, we believe there will be a conserved change at the epigenetic level that we can measure in the saliva. The term epigenetics refers to any trait that could be passed from one generation of cells or individuals to the next generation that does not rely on a change in DNA sequence for that transmission of the trait. The markers that we have chosen to study, we didn't even know existed 20 years ago. And it turns out that these are master regulators that don't rely on a change in DNA sequence to exert their effects, and yet they control the expression of most of the genes in the human genome. All the cells in your body can create proteins. The central dogma of molecular biology has held for over half a century that process involves going from DNA to RNA to protein. And we taught for many years that that's all you needed to know. We learned about 2 decades ago that there's a whole class of molecules that we refer to as non-coding RNA that strongly affect this whole process, going from DNA, RNA to protein. These are known as non-coding RNAs. They don't themselves code for proteins, but they are essential for this action to take place. There are specific classes of non-coding RNAs that are quite important in our test. These specific classes include microRNAs. MicroRNAs are made in all cells in the body. They actually are released by all cells in the body, and they travel in extracellular fluids like hormones throughout the body, and they can affect the process of making a protein in cells that are very distant from the cells in which they were originally made. In the case of our saliva test, we are looking at microRNAs, some of which have been made in brain cells and yet are found in the saliva. And that's one of the key reasons why this particular saliva test is providing a window into brain function. So the microbiome refers to the collection of microorganisms that live in a particular portion of the body or on a particular portion of the body that are quite distinct from the host cell or the host organism. And the interactions of those microorganisms with the host collectively forms a microbiome, like a mini atmospheric or small world in which lots of interactions take place. Machine learning that we used in the development of the saliva test enabled us to utilize 2 features of machine learning to our advantage. One of those is you can consider all possible components equally at the beginning. And when we talk about saliva, we had about 20,000 different features that could have contributed to the final molecular test. Through a machine learning algorithm, the important ones were narrowed down really by trial and error, a rote process that would take a human thousands of years to go through. The machine, literally the computer, can go through and test different combinations of features until it finds a test set that performs really well. Another critical component of the machine learning is the ability to take that test and evaluate it in a replication set, a holdout set, that was not involved in deciding what the important features were at the beginning. And that whole process of going through and selecting molecules you think would perform well in the test, evaluating them both in the training set and an independent test set, and doing that over and over again is what machine learning is really based on, regardless of the discipline in which you are using it. What is sensitivity? It's actually indicating the percent of times that you get an accurate result from the test when you have the actual condition that you're testing for. So some of the times when you use a test and you have the condition, you will get a false negative. So we see with the autism saliva test very high sensitivity. There's another metric that is used to evaluate diagnostic tests, and that's the specificity. When you have a result that indicates you do not have a condition, is that accurate or not? So specificity is really referring to the simple fact of when you do not have a disease or a condition, does the test actually report that result? Or does the test sometimes say you do have it? So that's specificity. When evaluating the performance of any molecular diagnostic test, you want to pay very careful attention to overall sensitivity and specificity. One of the statistical measures that combines both of those would be the overall accuracy of a test, or what we would refer to as the area under the curve in a receiver operating characteristic curve analysis. Early intervention through accurate referrals and accurate diagnoses is what this is all centered on achieving. The advantage of the saliva test from a pediatrician's perspective is that it will back up the opinion that they may have formed or been forming over the previous experience they've had with the child, and provide them a sound, objective, medically based, scientifically tested readout that can be used to confirm their suspicion. The saliva test takes a few seconds to obtain a sample and really a few weeks to get a full report that explains the results to that pediatrician. And we feel that that is something that they will want to have when they make the decision to either refer a child to a neurodevelopmental specialist for a formal, full-length behavioral evaluation or not. The saliva test, when it's properly applied, when it's properly interpreted, can be used as an aid to that process and really facilitate the initiation of appropriate therapies and interventions that can make a difference in the child's lifetime and in the parent's lifetime.